Now we discuss about the starvation response of bacteria in the soil. So, as you know, soil, if the bacteria is living in soil, it should be exposed to some kind of stress because of starvation due to lack of nutrient, or because of lack of moisture, they're exposed to some kind of drought. So whenever a bacteria starts its growth in soil, it will grow until either it is stopped by some kind of predator, or it is stopped by resources. As soon as the resources are vanished, as soon as the resources are finished, they will stop. Their growth will stop, or they will encounter some kind of predator, and the predator will reduce their population size to a limit where they will continue to grow as normal. So because in soil the bacteria are living in very sophisticated kind of environment in small pores, mostly predators have difficult access to these bacteria. Therefore, normally they encounter some kind of stress. So, and the soil-borne bacteria have ability to withstand the stress. Whenever they face some kind of stress, there is a reduction in the ribosomal RNA that shrinks. Then the proteins are degraded, some kind of contents of proteins are degraded. Then we see a series of genes are expressed, that these genes which are expressed in stress are known as starvation specific genes. So these genes actually increase stress tolerance. Then because of the gene expression, stress induced proteins are released, and then these bacteria can produce some kind of resting stages. If they can't, they are just die off. And it has been observed that only small fractions of resting stages can regain their growth upon onset of the normal conditions. So from here, we get the concept of dormancy. So dormancy means all the things are intact. The bacterial stage can regain its growth, but at the moment they are dormant. Their response doesn't start. The second is a condition where the starvation may take the cells to a stage where they cannot return to the normal growth. This we can say that it's a kind of death of the cell. So the two concepts are there: concept of dormancy and then concept of death in bacteria in the soil. So when we talk about the starvation response, some bacteria such as bacilli and clostridia, they produce internal spores that are known as endospores, as you can see in this figure. These endospores are highly resistant to extreme conditions like extreme temperature, radiation, pressure, and environmental stresses. They are the most resistant biological structures known. They are responsible for the persistence and survival of bacteria for many years, and they regain their growth when the normal conditions are on. Now, what are the methods, molecular methods that can be used to study the communities in the soil, especially bacterial communities? The molecular methods are mostly based on 16S RNA gene, which is used as a universal barcode for detection of bacteria. In this case, the whole DNA from the soil is extracted from the soil. Means all the DNA of all the organisms are extracted. And then we use specific primers by PCR, specific primers that can amplify specific regions in DNA that are specific to the bacteria or prokaryotic region. Then we can clone and sequence the amplified regions. The drawback of this technique is that, again, we extract, when we extract DNA, again, those bacteria which are bigger in size, they have more probability for extraction. Then they may be extracted better than the dwarf cells. Smaller the size, lesser is the cell lysis by different techniques. So ideal way is always to separate the cells based on size, as I have already discussed earlier in this lecture. So we can separate the sizes and then we can perform the extraction of community DNA. So, so far, by different molecular methods, by different molecular studies, we have observed that there is a big number of communities of bacteria or prokaryotes which have no representative or very few representative as cultured organism. So they have no cultivated representative. So this technique also support the idea that the huge number of bacteria in the soil that have their own DNA, they cannot be cultured. So these bacteria or these groups are have high abundance in many soil environments, and we see tremendous diversity within the taxonomic groups that were earlier established on the basis of cultivated organisms. And we see new groups, new subgroups within these established groups. So here, this chart gives you a small picture in which you can see the divisions of different groups, and each group contain non-cultured organisms. Very few cultivated examples are there. Now, there is another important group that I have separated to discuss here while I'm talking about prokaryotes in soil. So we should discuss about specifically about thread-like bacteria in soil, which are just like fungus, but they are not fungus basically. They are bacteria, they are single-celled, but they make colonies, thread-like colonies. When they divide, the cells remain attached. They are basically actinomycetes. As I have already explained, they are much more abundant in the soil. One species is example of one species. One example of species is Arthrobacter. Up to 67% abundance we see by this species, that is Arthrobacter species. Another type of bacteria that make chains are cyanobacteria. They make chains of cells along with heterocysts. Heterocysts, you may study about it. Heterocyst is actually a cell that is non-autotrophic. It's heterotrophic, but it gets its energy, its photosynthate, or its carbon from the other cells, which are actually autotrophic. Heterocysts are there in the colonies to fix nitrogen. So at the end, I would share the summary or take away points of this lecture, that culture bacteria are very small in numbers, but contribute most to the soil physiology and metabolism, first of all. Then the non-culturable bacteria are huge in number, but they are small in size. And we can say that they are small in size, so they are also known as the dwarf cells. However, they have a very small contribution towards physiology and metabolism of soil. There are different hypotheses to explain the non-culturable bacteria in the soil communities. One I already discussed in this lecture is that some people think that they are actually stressed cells of the same normal communities that we see in the culture form. Second community say that no, they are actually the cells of their own. They have their DNA intact, and whenever they are studied using molecular techniques, they show their their own groups, as we have already seen in this lecture. Then we discussed about the concept of dormancy and death. It's different from normal, as we have the concept in our normal life. In soil, we see number of cells which are dormant, and we expect them to resume their growth after a fixed number of years or fixed period of time. And their cells, we think that they may not resume their growth, so they are actually dead cells. Molecular studies have reflected huge numbers of non-culturable bacteria, along with the small numbers of culturable bacteria. The drawbacks of molecular techniques are also there, as we discussed in this lecture, that the extraction techniques are a bit biased or selective towards those who are bigger in size. Thanks very much.